In therapeutic development, proper nuclease selection is vital for safety, efficacy, delivery, and commercialization. But in a rapidly evolving landscape with regulatory roadblocks, this decision is more complicated than ever. Today, we'll hear from Casey Jowdy, Senior Product Manager at Synthigo, a specialist in strategic development and commercialization of CRISPR-based genome editing technologies. He will share in-depth data highlighting the cutting-edge performance, flexibility, and efficiency of Synthigo's engineered nuclease, eSpOT-ON, and HFCAS12 Max, along with how these key pieces comprise a platform that supports therapeutic development from RUO to GMP application. I'm James Crosspayne, your host with Cell and Gene. I'd like to thank you, the audience, for attending today's event. This is live, so we do encourage your participation. I'll be holding a Q&A with Casey following his presentation, so please share your questions with us using the Q&A box on your screen. Even if we can't address them all, I'll pass along any we miss for follow-up after the event. This question box can also connect you with me if you run into any trouble, but please try refreshing your browser first, as that will fix most common issues. Finally, today's event will be available on demand. You'll receive an email with a shareable link shortly after we conclude. Now, without further ado, let's begin today's presentation. Welcome, Casey.
Yeah, thank you for that intro. So let's get started. My background is in molecular genetics, and I've spent my career working at the intersection of genome engineering and product development, first as a bench scientist and now helping teams across research and translational spaces bring CRISPR tools into real world applications. And so today's focus is on two engineered nucleases, eSpOT-ON and HFCAS12 Max. I will highlight our portfolio approach from RUO to GMP solution that represent next gen of precision gene editing. So here's where we're going today. I'll start by unpacking why nuclease selection still limits translational therapeutic translation from both a scientific and operational standpoint. So then we'll dig into some eSpOT-ON data and a nuclease that's co-developed with AstraZeneca that's really pushing the boundaries of specificity and delivery compatibility in vivo. So for there, I'll introduce HFCAS12 Max, which we've had on market for about a year now. It's a high fidelity Cas12i variant. It's ideal for multiplex and hard to reach editing applications. And so finally, we'll wrap up with what looks like to scale from RUO to GMP and how our licensing and regulatory approach is designed to support you all the way to the clinic. So if you're currently using SP-Cas9 or Cas12a in your platform and thinking about what's next, this should give you a strong sense of where the fields and your options are going. So at Synthigo, we've supported researchers and therapeutic developers at every stage of the CRISPR journey. So from target discovery to ID-enabling studies. And across the landscape, one pain point that continues to come up, and that's nuclease selection. So a nuclease that performs well in a model system might fall apart in the clinical context, whether it's off-target activity, licensing entanglements, or delivery incompatibility. The wrong choice early on can really force teams to start over downstream. We've seen programs stall in the transition to GMP or spend months re-optimizing editing conditions with a new enzyme. So that's why our focus today is on solving that problem upstream. And we've built a toolbox that supports your entire clinical pipeline from RUO to GMP. And so with nucleases and synthetic guides designed for safety, scalability, and real-world delivery systems. And so Synthigo, founded in 2012 and started in 2015 supplying CRISPR reagents. By 2020, we extended that support into complete GMP manufacturing with this simple goal, and that's to help teams move faster with confidence and not compromise. So with that framing in mind, let's talk about what that right nuclease could look like. And so too often that conversation starts with what's most commonly used, but that doesn't mean it's optimized for your application. So instead, we need to ask more foundational questions like what minimizes rework and risk as you scale, what reduces off-target liability and simplifies licensing, and most critically, what gets you to the clinic without having to swap out tools halfway through. So we believe that answer starts with a platform that was designed, not just discovered, for therapeutic success. So you'll notice a little survey that pops up somewhere on your screen. We'd love for you to hear back from you. So while I'm giving the rest of this presentation, I would love for you to hear back and that survey. So we've had a lot of experience with customers using CRISPR in the clinic, and we've seen firsthand the negative impacts on suboptimal nuclease selection. And so repeating experiments, spending extra time optimizing, or worst case scenario, starting over completely with a new nuclease and a new target sequence. And so missing these regulatory deadlines and spending excessive time addressing regulatory feedback. This means delays, potentially years, likely more than doubling the costs and the risk that you may never get to the clinic. And so what we have done is partner. What did our partners do to successfully make it into the clinic? What they require in a nuclease is what features and characteristics did they prioritize that enabled them to successfully transition to the clinic? And so as an example of this, recently Synthego and Vita Therapeutics entered a non-exclusive licensing agreement with Synthego's high-fidelity HFCAS12 Max system, optimized guides to develop hypoimmunogenic master cell banks as a platform for advancing their iPSC-based cell therapies for neuromuscular disorders. And that's just one example, but we are expanding these options to include other engineered nucleases as well. So in the 5+ years we've been supporting CRISPR therapeutic development, Synthego has built out a comprehensive scalable platform, a platform that enables continuity from research use to GMP. And so this means same editing chemistry, quality control, same technical rigor from the very start, from the discovery stage, and carry you all the way through IND-enabling studies. And it's not just about having the right tools, it's about having a process that holds up to regulatory scrutiny and delivers consistent outcomes. And so whether you're using SPCas9 or HFCAS12 or our soon-to-launch eSpOT-ON, the goal is the same, and that's to power your team to build safe, effective, and scalable solutions. So let's start with eSpOT-ON, our lead engineered nuclease and a power alternative to SPCas9. So eSpOT-ON is engineered type 2B Cas9, and it was developed in close collaboration with AstraZeneca. The name itself stands for Engineered Specific Off-Target, On-Target, reflecting its dual design goals, so maintaining that high specificity of PsCas9 while enhancing the on-target performance. So PsCas9 is the natural predecessor, was originally identified from a human gut microbe species, and it's intrinsically high fidelity. So making it a promising starting point for therapeutic applications, but it really lacked robust activity on delivery constrained formats like mRNA and lipid nanoparticles. And so using structure guided engineering, based on cryo-EM data, Marcelo Maresca and his group at AstraZeneca introduced mutations and to strengthen the DNA binding, improve cleavage kinetics without really compromising specificity. And so the result is eSpOT-ON, really a therapeutic grade nuclease that performs reliably. And so recently at CRISPR Med 25 in Copenhagen, I'll paraphrase Marcelo and what he said in his talk there. He said, we didn't need another Cas9. We need one that could clear regulatory bars in vivo. And that meant fewer off targets, fewer translocations, and stronger on target activity in systems that I actually use in the clinic. And this is what AstraZeneca is using in their therapeutic gene editing platforms. And so, choosing eSpOT-ON is more about better editing. It's about de-risking your therapeutic program from day one. It's a forward-looking alternative for therapeutic developers who want that freedom to operate in high performance and a simplified path to the clinic. So before we move on into more specifics about eSpOT-ON and performance data, it's worth taking a moment just to compare eSpOT-ON to the standard SPCas9 enzyme. So both enzymes share the same PAM sequence as NGG and similar but not compatible guide RNA scaffolds. eSpOT-ON does stand apart in a few different ways, though. It's slightly larger, 1,400 or more amino acids versus 1,368 or so. And it is a type 2B system, and so not like type 2A, like SPCas9. The structural differences give eSpOT-ON, it's like a staggered cut pattern, cutting the target strand 3 nucleotides upstream of the PAM and non-target strand 6 to 7 nucleotides upstream. So which may lend to some advantage in repair pathways. SPCas9, by contrast, makes a blunt cut about 3 nucleotides upstream of the PAM on both strains. And so the full guide RNA of eSpOT-ON is a little slightly longer at 109 nucleotides versus 100 to 101. And this target sequence ideally based on some optimization to be a 22 base pair and not 20, which is most common with SPCas9. So the extended interactions may help drive its high specificity, while maintaining its high fidelity and cutting efficiency. And so while the delivery format and PAM may look a little different, under the hood, this enzyme is very different, but designed to cut differently, bind more precisely, and reduce that risk at the molecular level. So now we've seen what makes eSpOT-ON different at the molecular level. It's staggered cut pattern, extended target sequence, rationally engineered scaffold. The next question is, how does this translate into performance? So thanks to work published by AstraZeneca and their collaborators, we have a clear picture from the initial characterization of wild-type PSCas9 to the structure-guided engineering of eSpOT-ON. So the data really gains in specificity on target activity and in vivo relevance. And so these aren't just incremental improvements. They're meaningful advances for anyone developing CRISPR-based therapeutics. So let's dive into what the literature has to show us and see how the enzyme performs. So eSpOT-ON didn't emerge by accident, and it was a product of deliberate structure-guided engineering effort. And after solving the high-resolution cryo-EM structure of PSCas9, the AstraZeneca team identified key regions in the protein and sgRNA that could be optimized for better performance in therapeutic formats. And so on the guide RNA side, left-hand side there, that redesigned the scaffold to reduce structural complexity and improve the RNP stability. So that new V3 scaffold led to about 2.5-fold increase in activity. And on the protein side, 2 targeted mutations were introduced in the wedge domain. And so these changes enhanced DNA engagement at the PAM interface, improving overall nuclease activity up to 20-fold into delivery-constrained contexts. And so importantly, none of these changes came at the expense of specificity. Both change-seq and ddPCR assays confirm that eSpOT-ON maintains that high fidelity and low profile of the original PSCas9. So we'll get into that data a little bit further here. So, but what about its ability in cellular context? And so to address this dynamics, whether it performs consistently across cell types, not just simplified assays, but in cells that you actually intend to edit, they evaluated 5 distinct cell lines that are therapeutically relevant tissue types. So HEKs, HeLAs, HUH7s, DLD1s, and iPSCs. So in the study, both eSpOT-ON and SPCas9 were delivered in mRNA alongside their respective guide RNAs, and editing was assessed across 5 different genomic loci. And so what we see is a strong reproducible activity where eSpOT-ON across all 5 cell types, and notably it maintained high performance in iPSCs, which are notoriously sensitive and difficult to edit cell type. As you can see, performed as well or as better in many cases compared to the wild-type SPCas9. So it's also a direct answer to that central question of this presentation: does nuclease actually work in a therapeutic context? And I think the data from the publications and AZ's commitment to use eSpOT-ON in all of their therapeutic applications say that it clearly does. And even in other assays across a wide range of targets that I don't have time to go into, but are published in the literature, what we see is clear that engineered eSpOT-ON consistently outperforms wild-type PSCas9 and our approach to match editing levels of SPCas9 in nearly every case. So it's important to remember that SPCas9 has been optimized over the decades, and a new enzyme such as eSpOT-ON to clear the bar, let alone exceed under tight delivery conditions, is a strong indicator of translational potential. And so that data set was one of the first signs that eSpOT-ON was not just a safe choice, but also a high performance option for programs that can't afford to compromise with editing efficacy or fidelity. So to directly evaluate specificity, researchers used change-seq to compare off-target profiles across Cas9 variants. So wild-type SPCas9, Hi-Fi Cas9, that's marked in the bar there, and then from a commercial vendor, and then PS-Cas9, the unengineered version, and then eSpOT-ON on the bottom there. And so using this assay, it captures genome-wide cleavage events and reports them as a ratio of off-target to on-target reads. So these heat maps on the right on the top show 50 targets consistently detected across all 3 replicates, and each enzyme at 2 challenging loci, HEKS4 and TRAC. So wild-type SB-Cas9, as expected, show frequent off-target activity. High-fly Cas9 performs dramatically better, but eSpOT-ON demonstrates even the lowest off-target frequency across the board. And in many cases, off-target events weren't detected at all, represented here as those little gray tiles. And so this level of precision is critical, especially in therapeutic contexts and even in low-frequency off-targets, where low frequency off targets can pose a risk. And so with eSpOT-ON, the AstraZeneca team really achieved a meaningful reduction in off target editing without sacrificing that on target performance. So this in vivo study is designed to test eSpOT-ON's ability to mediate gene editing and therapeutic protein knockdown in mouse liver using LNPs as delivery. And so as shown in the panel E, the animal receives a single IV injection of mRNA encoding either eSpOT-ON in green or SB-Cas9 in light blue, and then buffer control in orange there, each targeting PCSK9. So in panel F, you'll see the results as eSpOT-ON achieved approximately 60% editing efficiency in liver tissue, substantially higher than SB-Cas9, which reached about 20%. And the buffer control, as expected, showed no editing. So panel G, the corresponding PCSK9 protein level in plasma. So the group treated eSpOT-ON green again showed the strongest knockdown with SB-Cas9 in light blue, yielding a moderate reduction and unchanged in the buffer controls. And so finally, panel H highlights a strong adverse correlation between the editing efficiency and plasma PCSK9 levels. So the higher the editing, the lower the expression, confirming this therapeutic potential of this approach. And so taken together, these data show that eSpOT-ON delivers potent genome editing and functionally meaningful protein suppression in vivo. So that's great. That was nice to see data showing high fidelity. The question is also about safety and tolerability. So on this slide, they evaluate some in vivo safety markers, liver enzyme levels, and body weight stability following treatment. So after a single IV dose of LNP formulated mRNA and guide RNAs, mice were monitored for signs of hepatic stress and systemic toxicity. So alternate ALT and AST enzyme levels shown here remained low and comparable across all treatment groups, including eSpOT-ON in green there, and SB-Cas9 in light blue, and the buffer controls in dark gray. So likewise, body weights remained stable over the course of the study with no signs of treatment-related decline or adverse effects. So when we combine these data with the 60% editing efficiency and a strong protein knockdown that we saw in the previous slide, it's clear that eSpOT-ON definitely delivers a pharmacologically relevant gene editing platform. So wrapping up on the section on eSpOT-ON, we can see that high-performance Cas enzyme engineered for therapeutic genome editing. It delivers strong on-target activity with minimal off-targets and translocations, as demonstrated in vivo efficacy. And so using mRNA and LNP delivery, to say the least, eSpOT-ON is clinically relevant. It's well characterized, and it's soon to be available exclusively from Synthego and mRNA, protein, and matched sgRNA formats. And in short, it's ready for your pipeline. And so, but we're not done there. We're going to explore a little bit about HF-Cas12, which we've had on market for about a year now, and how Synthego is building out our portfolio of Cas enzymes. So we've talked about eSpOT-ON. Our collaboration with Hutagene Therapeutics has been one of the most productive partnership in our CRISPR portfolio. And what emerged is HF-Cas12 Max. It's a purpose-built engineered version of Cas12i. It has an exceptional fidelity, a broad PAM flexibility, and a compact size ideal for delivery. And so Hutagene's team rigorously benchmarked it alongside Cas12a and other Cas12i variants. and HF-Cas12 Max consistently outperformed editing across editing efficiency and specificity. And so just like eSpOT-ON, we're not just offering another enzyme, we're offering a vetted, scalable solution co-developed with Futagene that they're already in the clinic with and have been producing lots of great data. And I'll just briefly highlight some of that data here. There has been several talks, and Futagene's been going around to multiple different conferences, sharing a lot of great data. And so I'll just highlight a little bit of that here. And so we've had HF-Cas12 Max for a little around a year now. It's a little bit smaller, about 20% smaller than SP-Cas9, which makes it ideal for delivery and via AAV and other size-limited systems. It also recognizes a 5' TTN or TN-PAM, shown on this heat map here in the green, benchmarked against other Cas12 variants, providing broad genomic access. And so the enzyme was evolved through a directed evolution strategy that targeted key DNA interaction domains, resulting in an editor that's both highly efficient and highly specific. And so it's been validated in multiple different formats, RNP formats, mRNA, and AAV, and it's shown excellent performance across T cells, liver, and CNS applications. So let's look a little bit at the data from HF-Cas12 in real therapeutic models. And so in primary T cells, they achieved 90% knockout of the TRAC locus and 80% viability post-editing. So that's not just efficient, it's well tolerated, which is critical for cell therapy workflows. And importantly, it demonstrates effective multiplexing and including simultaneous knockdowns of TRAC and B2M at the same time, consistently with high editing rates. And so once again, we also know about how, we want to know about how it performs in vivo. So we'll just take a look at some of that data here. And so using LNPs to deliver HF-Cas12 Max mRNA and guides, researchers achieved a significant reduction in cccDNA and HBsAg2 key markers of viral persistence. And this suggests real promise for curative interventions for chronic hepatitis B, where reducing the amount reservoir of infected cells has been historically difficult to do. And so importantly, this wasn't just effective, it was also safe. So mice tolerated the treatment well and no signs of liver toxicity or weight loss across the study, even after high dosing. And so this model underscores what we've seen in other systems. HF-Cas12 Max performs in vivo with clinical grade precision. And while not shown here, data from ALS models further reinforce this. Editing ATXN2 in TDP43 transgenic mice reduced neuroinflammation and toxic protein aggregation with meaningful functional improvements. And so combined with earlier T cell data, where we saw high TRAC knockout, strong viability, even with multiplex edits, HF-Cas12 Max continues to prove itself as a flexible and high fidelity platform across therapeutic success. And so building on this, Futagene's been very active in their therapeutic pipeline, advancing HF-Cas12 into multiple different clinical developments in their HD302 program, utilizing HF-Cas12 Max and DMD, and has entered the muscle clinical trial. So early data from the first dose patient indicates very favorable safety and tolerability with no serious adverse events reported. So moreover, initial assessments really show improvements in motor function and suggesting potentially therapeutic benefit. So as we transition from foundational data into the core of today's talk, it's worth pausing to look at the broader value of our engineered nuclease platform. So first, we lead with proven performance. These aren't theoretical enzymes. They've been validated in peer-reviewed studies across liver, immune, CNS models, and preclinical workflows that mirror real therapeutic applications. And second, we offer a simplified path to scale. So our licensing structure is designed to ease and lower upfront fees, clear milestones, and predictable terms that will let you plan with confidence. And so, and third, these tools deliver best-in-class editing with fewer trade-offs. So both together with eSpOT-ON and HF-Cas12 Max, they give you a flexible, high-performance toolkit backed by our transparent access. to these truly and true therapeutic intent. So as I briefly mentioned in the previous slide, let's talk about one more overlooked barrier, and that's clinical progress in late-stage licensing. And so we've seen companies pay 50 million for access to Cas12b and 75 million for a nuclease from another company just to unlock commercial rights late in development. So at Synthigo, we're taking a different approach. And so through our partnerships with AstraZeneca and Hutogene, we're offering sublicensable access to eSpOT-ON and HF-Cas12 Max with clear early stage licensing pathways. So this means you can run your preclinical programs without second guessing your platform, and you can plan ahead and not hedge your bets. And so because our terms are designed with developers in mind, the lower upfront fees, stage milestones, you avoid that sticker shock that often comes when the timelines are tightening. So this isn't just about smart business; it's critical for infrastructure for CRISPR therapy development. So we've covered the science, the data, and the licensing path. So now let's talk about what it takes to actually bring these tools into your pipeline from RUO to GMP and ultimately into the clinic. And so this is our approach to the problem, the CRISPR continuum, as we call it. And it's our answer to that foundational challenge. And how do you scale a research tool into the clinical product without introducing risk? And so at Synthigo, we built this pathway. We use same synthesis chemistry, guide format, same enzyme formats from across RUO, GMP. And the continuity reduces the variability, simplifies assay development, and streamlines regulatory documentation because you're not changing the underlying materials as you scale. So it saves time, it reduces batch-to-batch revalidation. It also eliminates one of the most common points of failure when transitioning from bench to IND. So it's not just about quality, it's about building with intent all the way from discovery to the clinic. So as a great example of our continuum with our recently launched GMP SPCas9 protein, which is available. If you're using SPCas9 and sgRNAs, Synthigo provides a holistic approach from RUO to GMP, meaning you can achieve all of those benefits I just highlighted for your SPCas9-based therapeutic. And so in addition, you'll benefit from manufacturing under full GMP compliance, under unified documentation, including full CMC support, drug master files available for both GMP SPCas9 and GMP synthetic guides, bundled pricing when you purchase all together, and consistency in specifications and quality attributes. And so this makes it easier for you to scale your platform, so reduce that validation burden and move faster towards the clinical milestones without changing the core of what's working. So let's clarify what makes our sgRNA and SPCas9 products like truly GMP, as we like to say. And so first, both comply fully with FDA and ICH CGMP regulations, meeting the strict CMC requirements essential for cell and gene therapy. Our production is governed by a robust quality management system certified under ISO 1344, ISO 9001. And importantly, we have a DMF for both GMP SPCas9 and GMP sgRNAs to simplify that regulatory submission. We also align with key regulatory frameworks, including 21 CFR, Part 210, 211, ICH guidelines, Q5, 7, 9, and 10. And every raw material comes qualified, QA-approved vendors, is fully traceable, and undergoes rigorous identity testing. And so our facilities include GMP certified Class C and D clean rooms, ISO qualified environments, and validated cleaning procedures, ensuring sterility, purity, and batch-to-batch consistency. And that's a lot. So finally, each lot is backed by full regulatory documentation, C of A's, TC/BSC compliance, and executed batch records. And so this is what we mean when we say true GMP, and that's how we empower teams to move forward without compromising on quality. Okay, so wrapping up here in the 5+ years that we've been supporting CRISPR-based therapeutic development, Synthigo has built out a comprehensive and scalable platform, a platform that enables continuity from RUO to GMP without compromise. And so whether you're using SPCas9 or ready to start with our best-in-class HF-Cas12 Max or eSpOT-ON, the goal is the same: we're empowering your team to build safe, effective, and scalable gene editing platforms on timelines that matter. But before we go, I do want to say a few words about where um, uh, what what we're about here at Synthigo, and that is behind every great product is a great team. And at Synthigo, we're intentional about how we build and support that team. And so our culture is rooted in servant leadership and strong retention, shaped by people who stay and grow and evolve with the company. We're not static. We iterate and adjust as we grow, and we stay aligned with the needs of our customers and with each other. So we prioritize people because we know that this leads to better outcomes for science, for business, and the communities that we serve. Also, communication and feedback are at the core of how we operate. We work across departments, we collaborate openly, and lead with clarity and accountability. And so this is how we're able to move fast without compromising quality. And we believe Synthigo is not just a great partner, but a great place to work. So that will we'll wrap up and we'll take some questions. But first, I do have some shout outs to give to a lot of key contributors here. I may not get to everybody, but I want to thank the team at AstraZeneca, Steve, Marcelo, Greg, Dima, Alex. They're a great team. I love to interact with them on the road and at conferences. They've engineered an outstanding nuclease with real potential in CRISPR-based therapies. Their work is really, truly making a difference. And also, huge shout out to Alvin and TJ Craddock at Hutagene, and to our internal champions at Synthigo, Kevin Holden, who's led the scientific direction of this effort, Adriana Zepeda for her pushing the program forward with all her great bench work, and Jason for his unwavering support, and of course my partner in crime, Mike here, who was instrumental in helping me build this and delivering this presentation for you guys today. And so I'm sure I missed some people, so thank you to everyone involved.
Thank you,
With
Casey.
that, I'll take any questions.
Yeah. Thank you, Casey, for that amazing presentation. Well highlighted how robust Synthigo's platform is for supporting cell and gene therapy development. Before the Q&A begins, I do want to remind our audience you can connect directly with Casey. His information is located on the side of your screen. And I see a ton of questions coming in, which we're very grateful for. I encourage you to continue submitting them via the Q&A box. So our first question is from Arav. He is asking, what is the length of eSpOT-ON guiding RNA, and are there modifications?
Yeah, so the guide RNA is a little bit longer in total overall length. It's 109 nucleotides. It's also 22 target 22 nucleotides in target length, so it's a little bit longer, but it is optimized and is shown to perform much better than some of the other sizes out there. And there are modifications to methyl and PS bonds on the ends. We tried out a whole bunch of different modifications, heavily modified, and really we landed on the most optimized modification pattern for eSpOT-ON.
Excellent, thank you for that. Our next question is from Donna, who is asking, what do you use to design gRNA eSpOT-ON for CRISPR experiments?
Yeah, so the guide RNA, there's not as much support as SPCas9, and it is a 22 base pair target sequence. So websites like ChopChop allow you to customize that target sequence, so you can get those NGG PAMs and that target sequence of 22 base pairs. Also, platforms like Benchling will also allow you to do more customized, and we're continuing to work with collaborators to bring more platforms in line with that, so you can use your favorite platform to design those guides, looking for off-target effects, etc.
Great, thank you so much. Our next question is from Felix, who wants to know how they can analyze their eSpOT-ON CRISPR edits after they've done their CRISPR experiments.
Yeah, so recently our partners EditCo are starting to host the ICE tool. If you're familiar with ICE, lots of people have used that platform for a long time. It's both eSpOT-ON, SPCas9, and HFCas12 Max are all supported on that ICE platform. So it uses Sanger sequencing, very cheap, very flexible, very robust platform that's been used by lots and lots of users. Of course, you could also use NGS, your favorite pipeline, if you're set up for NGS. But oftentimes, if you're doing a couple different edits, it's worth it just to do the Sanger sequencing. We have all the protocols there, and we're happy to support if you have any questions on how to analyze on-target effects.
Excellent. Thank you so much. The next question I have is from Erica, who wants to know if you will be offering GMP eSpOT-ON like you do for GMP SP-Cas9.
Yeah, that's in the works. It's not ready yet, but we plan on commercializing it as a GMP product, full GMP product with all the things I mentioned that we already have for SP-Cas9 on market now with all the regulatory DMF files, all that stuff. So that's all in the works. It's not quite ready yet, but we'll soon be at some point be announcing that. And I would imagine sooner rather than later because we're really excited about this product.
Great. Yeah. Awesome to know how many options there's going to be. Our next question is from Michael, who wants to know head to head why they should switch from SP-Cas9 to eSpOT-ON.
Yeah, so I mean, at Synthico, we certainly provide all of those options, as I mentioned, and how we built out our portfolio. I do think the purpose-built eSpOT-ON with its clear licensing pathway, still really high on-target activity. So you don't have to search for a lot of targets that are high efficiency. But just the safety and efficacy of it, the lower translocations and off-targeting effects, I think just makes it an ideal platform for therapeutic fans. So, and also, it's just a great research tool as well. It still has high on-target activity, very similar to SP-Cas9, as you saw from a lot of the work here. It's near as good or sometimes better in some locations. So I think the kind of the whole workflow and how you want to approach, I think is kind of that as you weigh the balance of your pipeline. So if you're just starting off, starting from scratch, then I think eSpOT-ON is, I think, a clear winner for a lot of different reasons, not only just from enzyme performance, but also licensing, regulatory, and safety.
Yeah, I think the presentation showed that really well, so I appreciate the additional context. So our next question is from Liana, who wants to know what the licensing path specifically looks like to use eSpOT-ON.
Yeah, that's something I think is probably best, in context with our sales representative. So best to reach out into our contact sales, and I think having a one-on-one conversation specifically around your therapeutic needs, goals, timelines, all those things, I think is. But we definitely have lower upfront fees, and we're really licensing this for developers. So that upfront, we're not just charging huge amounts, millions of dollars for access up front before you even get there, right? So we're working with you along the way so that you're successful then for that licensing platform. So I think this is a different approach, and I think this is the right approach for the cell and gene therapy field.
Great, thank you. We're getting a few questions actually from both Christian and Cecilia, who want to know how eSpOT-ON performs for HDR. Cecilia actually mentioning she spoke with Marcelo, I believe from AstraZeneca, who mentioned eSpOT-ON was more efficient at HDR than SP-Cas9.
Yeah, so there's possibly some advantages there with that staggered cut that lends itself to repair pathways that can make it more efficient. I don't have any data in this presentation, unfortunately, to show you, but hopefully there's as more data is published and possibly some stuff that we have coming out with several of our early access partners. I didn't mention that we have several groups that have already signed on, and they're putting this through the paces, right? So they are doing HDR experiments, and so we hope to have that application data for you. So hopefully soon, within the next 6 months to 9 months, and hopefully we'll be showing up at conferences and showing that data and to really answer that question more specifically for you. So with presentations and applications. So that's really a great question. I wish I had more data to show you right now.
Perfect. It's good to know that we have more to look forward to.
Yeah, absolutely.
So our next question is from Thasneem. I apologize if I mispronounced that. Who would like to know, you presented multiple options. Which one would you suggest as a better option over SP-Cas9, eSpOT-ON, or HF-Cas12 Max?
Yeah, so better is a relative term. right? It depends on their application. So SPCast or eSpOT-ON has an NGGPAM, and so if you've started to do some early work and there's locations with an NGGPAM that you can access with eSpOT-ON, that's has clear advantages, a little bit easier streamlined. But perhaps you don't have those, it's AT-rich regions, or you just need to get a little closer to specific target sites within the gene to make those knockouts. HFCAS12 is also an option that may be more specific to your particular case. So it's a little bit case by case variability. Also delivery format as well. HFCAS12 is a little bit smaller, so it can fit into AAVs a little bit easier. That smaller size does help it to get into some different delivery formats as well. So it is a relative type of approach, but fortunately we have solutions for both, and I think both enzymes are a great choice. I really think it just depends on the therapeutic application and target platform that you're going after. That really is going to lend itself to answer that question, what's best.
Excellent. Thank you so much. So I think we are going to wrap after just one more question. I want to thank the audience. We have so many questions coming in. I think some of them are a little bit more specific, so I'm going to pass them along to Casey and the team to follow up offline. So this question will be just a great reminder to end the presentation on. Tahera is both offering compliments, saying thank you for the great talk, and they want to know how they can get eSpOT-ON.
Yeah, so we'll be launching soon. I hope within the next month and a half. It'll be commercially available on our website. You can purchase those guide RNAs on our platform. Of course, reaching out to your sales representative. We'll have mRNA formats in multiple different tiers. So if you just want to get started with smaller sizes, or if you're ready to scale up into larger sizes, we have that benefit of those large scale sizes as well. mRNA, protein, and those matched guide RNAs that are exclusively available from us. Of course, you can take advantage of this offer that we have to buy some control guides that we have tested and vetted, that we know will work well, as well as non-targeting control and free eSpOT-ON. So 250 micrograms. So it's a good way to get started. Go to the contact sales form, fill that out, and we'll help you get started.
That sounds great. Thank you so much for that. And thank you, Casey, for a stellar presentation. It's been great.
Thank you very much. Thank you, everybody, for joining us.
Yeah, absolutely. We thank you, the audience, for being here today. And we hope to see you again soon. For now, hope you have a wonderful rest of your day. Take care, everybody.
Take care. Thanks. Bye-bye.